So if you now turn over, and we'll have a look now at how lipids are hydrolyzed and then absorbed. Now, lipids are slightly different because they exist in droplets. And lipids, obviously, we know are triglycerides, and we know triglycerides are hydrophobic, which means they don't like water molecules. Now, lipids, I'm just going to put here triglycerides because that's essentially what we're dealing with. And triglycerides, before we do any chemical digestion on them, we physically have to break them up, okay? So the first thing that happens is, within your pancreas, we produce bile, and bile contains bile salts. And bile salts emulsify lipids. So by being emulsified, they have a larger surface area. So emulsified lipids. And what we then hydrolyze them down to, we then add lipase, the enzyme, because once the lipids now have got a larger surface area, the lipase can then access the triglycerides and hydrolyze those ester bonds, and we form monoglycerides, and we also form fatty acids. And as you probably may have gathered, a monoglyceride is a glycerol backbone with a single fatty acid, and we make two fatty acids. So we're hydrolyzing two ester bonds there. And you can see here, you've got a large lipid droplet. There you have the addition of the bile salts, where you've got now a larger surface area, so you've now got many more small lipid droplets that then the lipase can work, and we can then hydrolyze those to fatty acids and monoglycerides. And fatty acids. Make sure the colour's okay. So now if we practice writing that out in sentences, so we can see why we're doing that. So now we've got the reasoning behind that. So bile emulsifies, that's a nice key word there, large lipid droplets into smaller lipid droplets. What I'm going to do now is say why that we need that to happen. So what's the purpose of that? This increases the surface area for lipase action. So why is that an advantage? And I'll do that in a different colour. This makes the digestion of lipids much faster. So then what does lipase do? So lipase hydrolyzes triglycerides into monoglycerides and fatty acids. Okay, so that's where we are now. So you've got, that's the large into smaller, okay? Now then what happens, if you imagine then what we said before about triglycerides being hydrophobic and not wanting to interact with water. Well, if you think about monoglycerides and fatty acids, they're the components of a triglyceride, so they'll behave in a similar way. So they're also hydrophobic. And in order to overcome that, what we need to also do is find a way to be able to transport them to the cell to enter the cell. Now, they're still hydrophobic. So what happens is the bile salts actually form a temporary structure with the monoglycerides and the fatty acids. Now, the bile salts themselves have hydrophilic parts and also hydrophobic parts, which means they can interact both with the monoglycerides and fatty acids on the hydrophobic part, but they can also interact with water on the hydrophilic part. Now, this structure here is called a micelle, and that is the structure that allows the transport of the monoglycerides and fatty acids to the membrane of the epithelial cell. So, micelles are formed which are composed of monoglycerides and fatty acids associated with bile salts. If we didn't have the micelle formed, we wouldn't be able to transport those monoglycerides and fatty acids to the cell membrane because they still are hydrophobic. But having them in the micelle allows that to happen. Now, what happens is the micelle will move towards the membrane. Now, the micelle itself is lipid soluble, so that means that it can transport inside the cell or diffuse inside the cell and carry with it the monoglycerides and fatty acids. So the micelle carry the monoglycerides and fatty acids across the phospholipid bilayer. By simple diffusion as they are. I'm just going to change the colour because it's an important point we need to make. They are lipid soluble. Okay, so then what happens is, imagine now this is where you are. So you've just been transported into the cell, and I'm just going to draw on this diagram here so you can see. You've now got monoglycerides and fatty acids. Now, what then happens is the monoglycerides and fatty acids enter this structure. If you can guess what that is the SER, or the smooth endoplasmic reticulum. And we know that the role of the smooth endoplasmic reticulum is to synthesize lipids. So the monoglycerides and fatty acids enter the smooth endoplasmic reticulum, and what happens there is they're resynthesized back into triglycerides and reform into triglycerides. Okay. So then what happens is, we know in the cell as well, if you want to add this on, because we need this structure, we've also got an ARIA, or a rough endoplasmic reticulum, which makes proteins. So then what happens is proteins are formed in the rough endoplasmic reticulum. Now, we know proteins get modified in the Golgi apparatus, or the Golgi body, okay? So then, what happens here is the triglycerides and proteins, along with cholesterol that's made in the smooth endoplasmic reticulum, and phospholipids, are modified and assembled in the Golgi apparatus. So we're combining them all together. Now, once they're all combined, so the combination is made of triglycerides and proteins with cholesterol and phospholipids. This forms a structure called a chylomicron. So forming chylomicrons. Now, when we form the chylomicron, what happens then is, and you can see that on this bit on the diagram, so we've now formed this chylomicron, and what they are made of these different molecules here. Now, the chylomicron just move this bit over here, still see the diagram. The chylomicron is released from the cell, by exocytosis, where it enters, now this mound at the bottom here, the lacteal, which is where it will become lymph before draining to the blood. And that is the digestion, which is this bit up here, and the absorption of lipids.